we know energy rich molecules. So what are energy rich molecules? NADH, FADH2, all these are energy rich molecules which are produced so in various pathways like glycolysis, it is produced in TCA or even fatty acid oxidation. So all these pathways, these energy rich molecules are produced or formed, and these energy rich molecules, so gets transferred from the biomolecules. When it gets oxidized, ultimately, so it enters the ATC, in the electron transport chain. So there we can see a flow of electrons through a set of electron carriers, and ultimately it passes electron to the molecular oxygen. So what do you understand from oxidative phosphorylation? So oxidative phosphorylation is a process in which ATP is formed, so as a result of transfer of electrons from the energy rich molecules NADH or FADH to transfer of electrons finally to molecular oxygen, so through a set of electron carriers. So the process of synthesis or the production of ATP, so when these electrons are transferred from the, that means energy rich molecules to the oxygen, we call it as oxidative phosphorylation. Where it takes place, where is the location? Inner mitochondrial membrane. So process takes place in the mitochondria, inner mitochondrial membrane. Here definition is very important. What is oxidative phosphorylation? Process by which ATP is formed when electrons are transferred from energy rich molecules to oxygen through the transfer of electrons. So that process we call it as oxidative phosphorylation. So what are the steps so involved in, or what are the components involved in oxidative phosphorylation? There are two components. It includes electron transport chain as well as ATP synthase enzyme. Okay. So here, if you see step by step, so this is the flow, like the flow of electrons from the reducing equivalents. Reducing equivalents are nothing but NADH and FADH2. So which transports the electrons. So and when it transports the electrons, it is involved in generating the proton motive force. So this energy which is stored in the proton motive force is used to drive the synthesis of ATP by activating the enzyme. Which enzyme? ATP synthase. So oxidative phosphorylation, it includes two different components. One is the process that takes place in the electron transport chain. The other one is through the proton motive force, it activates the ATP synthase. That is why it is involved in production of ATP. So coming to the chemi-osmotic theory. So we call it as Mitchell hypothesis. So what we call it as chemi-osmotic hypothesis. So this is the most widely accepted mechanism. So for which is proposed for understanding the mechanism of oxidative phosphorylation. So I'll just make it simple. No need to read this. Just see the diagram. The diagrammatic representation is very important. When you see the diagrammatic representation, so one side, so it is a cytosolic site, intermembrane space. On the other side we have, that is mitochondrial matrix. So when the electrons, these are complexes, different complexes which are involved in electron transport chain. Complex 1, 2, 3, 4 and all these things. And this is the enzyme complex for ATP synthesis. Otherwise we call it as F0, F1 component. Right? That is the component. So what happened here? So during the electron transfer, so that means via this energy rich molecules. So there it accepts the electron, and it is when it undergoes simultaneous oxidation and reduction, it passes electron from one set of electron carrier to the other set of electron carrier. That means electron moves from one complex to the another complex. So there is a flow of electron. So during the flow of electron, simultaneously the phosphorylation also takes place. How this phosphorylation takes place, that is the mechanism of oxidative phosphorylation. Because when it transfers the electron at particular complexes, for example complex 1, not complex 2, complex 3 and complex 4, complex 1, complex 3 and complex 4, these are the three specific sites where ATP is formed. How ATP is formed? During the electron transfer, it is coupled with the proton transfer. Proton transfer from where? From the matrix to the cytosolic site. When it pumps the H plus ion, that is why we discussed it. So in complex 1, it serves as proton pump. Whereas complex 2, it doesn't serve as proton pump. That's why there is no ATP formation. Whereas in complex 3 and complex 4, again it serves as a proton pump. That means, so during the transfer of electron, so simultaneously it pumps the H plus ion from mitochondrial matrix to the cytosolic site, that is intermembrane space. So ultimately what happened? It results in increasing the potential on the other side. It creates the electrochemical potential. There is a difference. Okay. So initially it is positive in the matrix, and slowly due to the pumping of the H plus ion, so the H plus concentration is, that means, increased on the other side, that is in intermembrane space. So this increased H plus ion, that means proton gradient. So this proton motive force, this force we call it as, considered as proton motive force, and when it creates the electrochemical potential over here, this increased H plus ion concentration, so it activates, it comes and binds with this protein, that is ATP synthase. So it activates this ATP synthase. So increased H plus ion, it binds at the specific sites and it activates the ATP synthase. So, and thereby it phosphorylates the ADP and it results in formation of ATP. So this is the basic mechanism which was proposed by Mitchell, and we call it as chemi-osmotic hypothesis. So here what we have to understand, key point is that, so during the electron transport, it is coupled with proton transfer. Right, this gradient mechanism, so the proton gradient mechanism which activates the enzyme, so it is involved in formation of ATP by phosphorylating the ADP. Right, so this is the mechanism of oxidative phosphorylation. So we call it as chemi-osmotic hypothesis. They might ask, so describe the chemi-osmotic mechanism of, that means the oxidative phosphorylation. This diagrammatic representation is very important, and we need to understand, so the H plus ion concentration and why ATP is formed at that particular site. Okay, why ATP is not formed in this particular site? Because free energy potential is not satisfied at this particular site, so the proton transfer will not take place even though electron flow happens. Whereas these specific sites, so electron flow at the same time it pumps the H plus ion, because there the free energy potential is satisfied at those specific sites. So it activates this particular enzyme, so that phosphorylates the ADP, which is involved in, that means, formation of ATP or production of ATP. So this mechanism is very, very important, mechanism of oxidative phosphorylation. So in general, what we understand what is oxidative